So now I am gonna hand the microphone over to David Leclerc. So David, please take it away.
All right. Well, thank you so much. And thank you for coming. This is the second, I think, or third time we are hosting a webinar for our friends at Curiox. And the technology is not specifically flow cytometry, so I thought I'd give a few words to explain what exactly we're thinking about here. Essentially, we're looking for expanding our services or exploring how we can expand our services and offering a service that would help our users better prepare their samples. So there's essentially
David, sorry to speak over you. You just went muted.
all right. Where did I cut off?
You talked about improving sample prep.
Okay, improving sample prep. So essentially, there are two questions we're thinking about when we uh, look at the type of technologies such as the one that Curiox is offering. The first one is, can we improve the sample quality? So flow cytometry is involved in a variety of experimental pipeline, and so very often the cells that will come to our car will end up in other core facilities such as sequencing and so on. And there's a bunch of steps that we can take in our facility to better prepare the samples so that at the end of the experimental pipeline, the data that gets generated is of higher quality. So essentially, you get more bang for your bucks if you go through our core facility. So uh, that may include technologies that will help you remove debris or dead cells from your samples, or any technology that will help us keep the viability of the cells at the highest level throughout the course of your experiment. So while you stain your cells, while you process them through a cell sorter, all of that will help us, or keeping the cells at the high viability will help you get better quality data at the end of your experiment. Um, the second question we have is, how do we improve uh, the quality? I keep looking at my microphone. I'm paranoid now. But the question is, how can we improve the workflow? Um, is there a way that we can essentially improving the workflow will include automation? And for those of you who've looked at the latest acquisition in our facility, all the benchtop instrument that we've made available lately have all offered a really reliable auto sampler that will allow you to pick up your samples from a plate. Um, and so essentially it allows you to come to their facility, put your put your samples on the machine, and kind of walk away while the acquisition takes place. And so now that we have that available, can we expand that to sample preparation? If you're going to prepare your samples and put them in a plate, can we help you prepare your plate of samples? And we're talking here about staining your cells, fixing, and so on. And so down the line, our hope is that this is going to be something we'll be able to help. It will provide you some cost savings. So just not having a technician needed to um, be there feeding tubes or pipetting antibodies to a random series of Eppendorf tubes, that just there saves you quite a bit of time. So these are the type of features that we're going to be looking for in technologies offered by the Curiox. So this our call to action at this point is simply to get in touch with OE, with Anne, try to get your hands on the instrument, test it out, and uh, come back to us. Let us know how that went, and let us know if you feel this is a technology that we should be offering in our facility. So with that, I think I'll turn it back to OE and Anne.
David, thank you very much for that overview. And in listening to you, you know, the two goals that you highlighted really resonate at the core of what Curiox Biosystems stands for. You know, number one, the improval of sample prep. Number two, the improval of the workflow. And really, for the next few slides, myself and my colleague Anne would like to really continue that conversation on the benefits of automating your sample prep process by introducing you guys, our audience, to a novel and exciting technology that was specifically designed to improve your sample prep called Laminar Wash. Now, first of all, why do we need something new and improved? In short, the answer is the old ways are getting outdated. Science has come a long way. You know, the precision and advancement of next generation analytical tools, such as multi-parameter cytometry, single cell sequencing, high throughput research analysis has allowed us to unlock very impactful applications of our research, pretty much
Hi,
enabling
Ogi.
us. Yes.
I'm sorry to interrupt you. I can't see your screen.
Aha. Thank you for letting me know that. Let's try this again. How about now? Can you guys see my screen?
Yep, looks good.
Great. Thank you very much, and I appreciate that. So as I was saying, you know, we can do a whole lot more with a lot less because of advanced technology. But a key bottleneck that remains in this process is the sample preparation. You know, we still rely on a manual process that is either time consuming, variation among users can often impact data consistency and reliability. Our main way of washing ourselves involves centrifugation and aspiration, which can often be harsh on cells, especially when you're dealing with precious or fragile cell samples, which can then impact viability and even cell loss. So which brings the question, you know, what if we can do better? I'll rephrase that. Let's do better. And that's the idea behind Laminar Wash. You see, our Laminar Wash system was designed to streamline the sample preparation process and introduce benefits such as increased accuracy, faster handling, more reproducibility, making it gentle on your cells, more time savings by minimizing the hands-on time that the user has to engage with the samples, just to name a few. Now, what are some of the benefits that Laminar Wash has brought to sample prep? Well, first of all, by avoiding the harsh pelleting of cells induced by centrifugation and aspiration, we are able to recover more cells. So this was some data collected from the Nenki Institute Flow Lab in Sweden, where they compared both the number of lymphocytes and the percentage of viable lymphocytes. And they observed that by using centrifugation, by using Laminar Wash compared to centrifugation, you get better samples to start with. Now, another group, Proteona, compared the quality of recovery, comparing Laminar Wash and centrifuge at different cell inputs. And what they observed that with Laminar Wash, you get better recovery, especially when you're working with a low number of starting material. Laminar Wash also allows reduced hands-on time, which means by enabling faster washes. With the Laminar Wash system, each cycle of wash takes, roughly speaking, about 25 seconds per wash cycle, which allows you to streamline that process with minimal user intervention. And then thirdly, the automated system is not dependent on the sample handling ability of your user or the employee or the technician, meaning you have less variation and you have more reproducibility across replicates, across users, across time and space, providing you the peace of mind on the quality and reliability of your data. So now that's just in general. Let's take this to flow cytometry. You know, flow cytometry is a great technology. We've been able to do a lot with it. However, it does come with its own challenges. For example, if you're working with primary samples For example, tumor microenvironment analysis. Debris removal from those samples is key. You know, flow cytometry often requires, a labor-intensive washing and staining step, which, depending on the user, can be slow and inconsistent. And then there is often a need to retain viable cells, especially if you're working with rare populations and rare events. Now, when you're using the conventional centrifugation-based washing, well, it relies on the user's technique for either decanting the tube or flicking the plate, which can vary. Centrifugation cannot discriminate between dead cells and live cells. Oftentimes, centrifuging pellets debris as well as dead cells in addition to your cells of interest. And its subsequent spin, centrifuge, and aspiration risk more cell loss and compromises the viability. Well, with laminar wash, we propose something different. We want to introduce a gentle washing of these fragile and rare cells. We want to efficiently remove debris, dead cells, and any unbound reagents. We want to minimize the hands-on time to where the user spends interacting with the samples, and we want to ensure that your gating is standardized and your analysis is reproducible every time. So I'd like to show you just a case study of that. This was a group based in Boston that were interested in studying the tumor-infiltrated lymphocytes from a mouse tumor model. They introduced tumors into a colon cancer cell tumors into a BLBC mice, generated the tumors, isolated it, and then generated a single cell suspension with the goal of characterizing the immune cell populations within the tumor. And they did this using laminar wash compared to the standard centrifugation process. And immediately, some of their their striking observations were with laminar wash, comparing laminar wash to their manual process, they saw increased viability, but not just increased viability. If you look at their forward and side scatter gating, they observed that in that corner where debris usually collects, laminar wash was able to reduce the amount of contaminating debris in their sample, meaning they were able to enrich for more of their cells of interest. Also, as I mentioned earlier, more live cells, better population resolution, meaning better gating strategies for them. And here's just some feedback that we received from the lead scientist of that project. And he commented on increasing the workflow efficiency, increasing data reproducibility, data consistency, better cell retention, better cell viability, better resolution of cell populations, which is key if there are those hard to define or hard to capture populations. We were able to minimize stress on their sample, eliminate better clean up debris and autofluorescence from the background, minimize clumping, which ensured that their cytometer was less chance of the cytometer clogging, lower single cell suspension, and overall just better management of their sample volumes. Now, we're also glad to share that University of Chicago actually recently benefited from the laminar wash system as well. See, in 2021, just last year, Dr. Shireen Rouhani recently published a Nature paper using our laminar wash system on the on COVID-19 immunology study. And you can see here's what she had to say. The main point I want to really capture from her statement was the laminar wash technology allows you sample staining for flow cytometry completely within a BSL-2 cabinet without centrifugation to work safely with this infectious sample. So not only do they get better handling, but for those that are working with contagious or whether BSL-2 samples, you have that peace of mind of better biohazard containment. So the question that people are always asking when we get to this point is, well, how does the laminar wash system work? Well, I'm glad you asked. See, first we have we have a patented laminar wash plate uniquely designed with a hydrophilic well surrounded by a hydrophobic surface to hold your samples. This allows users to reap all the benefits of sample minimization, since each well holds up to 5 million cells in a minimal volume of 80 microliters, and without compromising the efficiency of staining. For those that need a larger volume, we do have a large volume adapter capable of accommodating up to 150 microliters. When the samples are loaded to the wells, the cells settle naturally without any external force. This settling step can be combined with antibody incubation for efficient staining, especially for flow cytometry. Now, due to the difference in cell density, the live, intact, and healthy cells will settle to the bottom, while unbound antibodies, cell debris, and dead cells remain buoyant at the top of the well. After the settling process, the user loads the plate into the washer, and then they can select their customizable flow rate and number of washes, and these are customizable based on the needs of the experiment. The system will load the fluidic head onto the well and initiates the laminar wash. And what really happens is you have two nozzles in that well. One nozzle dispenses fresh buffer, while the other nozzle aspirates waste. And by repeating this, we generate a laminar flow that ensures optimal removal of the buoyant debris, dead cell, and unbound reagent. Meanwhile, your target cells and your healthy cells of interest remain undisturbed at the bottom of the well. And so afterwards, the cells can be taken out for further uses, or the plate can be loaded onto most cytometers with 96 well plate reading capabilities for direct reading from the plate. Now, I know the goal of we're working with the flow core at University of Chicago, but as David pointed out earlier, because the samples that go from flow are applicable to a variety of uses, I'd like to share with you that the laminar wash system actually has a broad utility ranging from not just flow, but to single cell sequencing, mass cytometry, multiplex immunoassays, immunotherapy. What I like to tell people is, if you don't see your application on this list, please reach out to us, because there's a good chance that if you're working with cells, we could benefit the work that you are doing. To just sort of wrap up my part of the laminar wash, just like to share with you that Curiox Biosystems offers a suite of laminar wash solutions to meet a variety of user needs. We have our laminar wash mini for low throughput samples, capable of washing up to 8 wells at a time. And this is generally suited for use in individual labs that don't have high throughput analysis or samples that they generally work with. Then next, we have the AUTO1000 for higher throughput preparation, and it is capable of washing and staining 196 well plate at a time. And lastly, the AUTO1000, which my colleague Ann will give us some information in the next slides, is a fully automated hands-free sample prep station of up to 296 well plates at a time. So at this point, I am going to pass the baton to my colleague Ann. And guys, remember, if you have any questions, please feel free to type it in the Q&A chat box. So at this point, I will stop sharing screen, and Ann, this floor is yours.
Thank you, Ohi. Let me just figure out how to share this. Okay, can everybody see my screen?
I can, yes.
Okay, fantastic. All right, thanks, Ohi. Let's see, okay, there we go. So thank you for being here. Good afternoon, everyone. So like Ohi mentioned, my name is Anne. I'm the automation manager here at Curiox. And today I would like to tell you just a little bit about the AUTO1000 system that we have. So Ohi has already familiarized you with the Laminar wash technology. The AUTO1000 is our largest product. It is a fully automated cell processing workstation. As you can see on the left here in the screen, we have pull up my laser pointer. This is where the AUTO1000 washer will sit. And there is also a buffer exchanger that sits underneath the washer. And then we put the washer in an entire platform of a Hamilton Nimbus extended. This entire platform will run one assay at a time, but it can do 2 plates consecutively in total. There's also a HEPA filter, which is positive pressure, which is optional, but available. So this entire platform works on a 4-channel system. So there are 4 pipetting channels that actually usually park here. Each of those channels can hold up to a maximum of 300 microliters. So the tips actually go here. And these are conductive tips. So they can actually measure and sense how much liquid is in a well. And that really increases the precision and accuracy of pipetting as this entire system handles liquids. There are also 2 grippers, which are parked here normally. And so these are the 2, they look like little hands, but they are what the Hamilton Nimbus platform uses to pick up plates and move them around the deck. The startup for this entire platform is pretty fast. It takes about 25 to 30 minutes in the morning to start up. And as Ohi mentioned, this platform is capable of doing anything here on the right. As you can see, we can do viability stains, surface stains, fixed perm, intracellular stains, sample washes of your cells, and even a buffer exchange or a final transfer into an output plate, or you can keep it on the Laminar wash plate and read it directly in a cytometer like Ohi mentioned. See, all right. So here's the key component of the AUTO1000. We've actually developed a user-friendly graphical user interface that enables labs to fully automate without the need to script and code in-house. So this means that this GUI here, as we call it, is designed for scientists to use. So you don't need to have an engineering degree. You don't need to go through any programming in order to learn how to use the entire platform. This is just a screenshot of what this looks like. I will show you more at the very end. I'll go through a little example of the GUI so you can see how it is easy to use. This entire platform, like we mentioned before, is designed to be a turnkey walkaway solution. So basically, you just set up the system, enter your protocol using the graphical user interface, press start, and you can walk away and come back when it's done. This is the most useful to fully automate a repetitive experiment that really demands precision. So most cell analysis would work like that. So you can do staining, you can do a lot of types of processing on this platform. There we go. So I'd like to show you a little bit of data on this because the proof is in the pudding. So here is another case study that we did actually in conjunction with our collaborator over at Charles River, which is just down the street in Worcester from our office in Boston. So there were actually 5 animals that we processed here. All 5 of them look about the same. So we picked 3 to show on the screen. You can see that the Laminar wash platform and the SOP yielded about similar results. So the manual method, which is the conventional SOP with the centrifugation, flicking, adding more antibodies, etc., is shown in gray. So these are the gray bars that you can see. And the AUTO1000 is shown in blue. So we did 3 replicates for all of the manual methods and 4 replicates on the AUTO1000. And you can see that all the bars, the blue bars are much smaller, the error bars are much smaller, and the CVs are much lower with the AUTO1000 platform. This shouldn't come as a surprise to anybody who works with automation, because essentially machine does the exact same thing over and over again, whereas a human can be influenced, understandably, by many other factors. You can see that the 2 methods actually yield pretty similar subpopulation results, but the key difference here is really in the reproducibility in the CVs that we see here. This is, I should mention, just some ACK-lyzed blood from humanized mice. And these are the results that we see consistently with a lot of other of our customers who are using either automation or the AUTO1000 platform. So here's a little bit more flow data. So this is done on a different cancer model. We are looking for TILs in this particular case in humanized mice, again, with our collaborator. So here you can see clearly in the first column. The top row is actually cells processed in the conventional method with a centrifuge and flicking and adding more reagents. And the bottom is with laminar wash. So in this case, just laminar wash itself. You can see in the front panel, the first panel on the left, that it's really clear that the debris removal is much better on the laminar wash platform. This is in part due to the fact that it is very easy for us to do more washes, and it comes at no detriment to the cells. So we're able to wash as many times or as few times as we want. And so this really helps to get rid of the debris, which for some of you, if you work with tumor samples or any kind of dissociated tissue, you'll know that debris is a really big problem in processing these types of samples. You can also see in the middle panel here, here's a live dead stain, that viability ends up being much higher at about 51% versus about 37% between laminar wash and the centrifuge. And again, this will make sense intuitively if you understand the platform and what high G forces can do to cells. A lot of times these G forces really are crushing the cells, and a lot of them end up, so in a lot of samples, you end up seeing lower viability. On the laminar wash platform, because it is so much more gentle, we tend to see higher viability in this case. And the last thing I want to draw your attention to are the two panels on the right, CD45 and CD3 stains. You can see that because there is less debris, the population resolution is actually much better. So for those of you who may use an automatic gating strategy, this will be very helpful as well. We have seen some customers who do that report much better results with using the laminar wash platform because the population resolution is so much better. All right, so just to summarize a little bit before we move on to the GUI, I hope we've been able to communicate in just a really short amount of time how laminar wash can bring workflow improvements to your lab in terms of time savings and hands-on time and the ability for you to walk away while the assay is going on. In terms of consistency and reproducibility, so the results you get will be independent of operator and independent of location or time of day or night. We've hopefully been able to prove as well the biosafety component in that the system generates no aerosols at all, like centrifugation can if you flick your plate. And the laminar wash platform can fit in a biosafety cabinet, and the AUTO1000 is enclosed entirely. As well, you can also see, we can expect to see higher quality data by maintaining the cells in their native state. And this typically will help improve retention and viability and maintain cellular morphology. This can also aid in debris removal so that we're getting rid of all the junk that you don't want so we can get to the cells that you do want. This will also help with cell washing in generating clearer resolution of distinct populations and perhaps in elucidating rare cell populations. So with that, I would like to, let me stop share for a minute while I pull up another demonstration for you. Okay. So any questions while I pull this up in the meantime? Oops. All right. I think you can see my screen now. This is just a simulation that I'm running. Everybody can see this?
Yes.
Okay, fantastic. Alrighty, so I've just pulled up a simulation of the graphical user interface. So when you first launched a program, this is the first screen. This is the launch screen that you would see. So you can see our logo at the top, and this is actually a tiny mini cartoon of the washer by itself. So from the screen, you can execute a daily startup maintenance and even perhaps a calibration. So you don't have to actually stick your hand into the machine to run all the startup maintenances. You can also run a protocol, which we'll do in a little bit. We'll set up a protocol together. And at the end of the day, you can also execute all of your shutdown maintenances from here. So let's go ahead and enter a protocol. So if you hit run protocol, you have two choices. Either you can configure a new run, which is what we'll do today, or you can load a saved run. So here you can go and browse, and it will pull up all the protocols you've saved before. And so if you have a protocol that you run all the time, you can save it and then just load that every day without having to go through this process. But if you're doing a new run, let's go ahead and do that. So we have a choice of 1 to 2 input plates. Let's go ahead and select 1. You can also select 2. Like we said before, the system is capable of running 2 plates in total, but it will do 1 assay at a time. Let's go back to 1. All right, if you hit continue, this on the top right here, you'll see a loop number. Every plate gets up to 10 loops. You don't have to use all 10 loops, but you do get the choice of up to 10 loops. Every loop is organized as such. The logic that we have built for this is based off of most cytometry protocols, which is some kind of addition, some kind of incubation, and then some kind of wash. If you think about most protocols that you've worked with, either in flow or in mass, in my cytometry, you will usually add something, wait a little bit for it to incubate, and then wash it off, and then add something else, wait a little bit, wash it off. And so this process repeats over and over again, which is why we've designed the loops to be like that. So the first step is always a sample transfer. So let's pretend today that we are doing 24 samples with 2 replicates. So you can do up to 4 replicates today. Let's just pretend and go ahead and say that we're going to do 2. This will fill up half of a plate. You can do anything. You can go as low as 1 sample and just do 1 well, or you can do a full 96 well plate here. We can select how much volume we want to transfer. So for example, I'm going to do 50. And let's say I want the cells to settle for 30 minutes at fridge temperature. So I'll select a 30-minute incubation time and a temperature of 4 degrees. So you can select anywhere from a 4 to 70 degree temperature range. Most of our customers will do room temperature or fridge, so 4 degrees Celsius. And then we can wash 9 times. So as Ohi mentioned before, these washes are not synonymous with centrifuge washes. Because every wash, even at the slowest possible speed we can set the system to, every quote unquote wash will only take about 40 seconds. So a 9-time wash will take you about 5 minutes. And this is at the slowest flow rate possible. We typically set the default to 10, and you can select a wash buffer. We can just leave this where it is. And let's say I want to vortex the plate after washing. So this step, I'll just explain a little bit, is with, you've heard Ohi talk about the cells settling to the bottom of the well before we wash it. So when the cells have settled, they've been washed, but after the wash, they're still settled. We haven't really disturbed them, but we want them to mix with whatever reagent is coming next. And so we vortex the plate to lift them off the bottom of the well. So once we selected that, we can just confirm this particular step. Everything looks good here, so I can go ahead and hit continue. All right, I'll ask if you want another assay step. In this case, yes, we do. All right, so top right-hand corner, you'll see loop number again. This is loop number 2. You'll also see the number of samples and number of replicates here. I don't know about you, but I sometimes forget how many samples I have. So this is a good reminder right here. So every other loop, loops 2 through 10, if you do use all 10, but every other loop from here on out looks the same because you are just selecting the number of panels to go into a well. So let's say I will add 50 microliters of my antibody into the well into my 24 samples, and I want it to pipette mix after a transfer. So here is where you get to have some fun. You have up to 10 panels to play with for every plate. So I can say, let's say I want, in this case, 23 all stain samples and maybe 1 unstained control. You can do that here. If you want all your samples to get the same panel, you can also do that here. If you would like to split your samples and do 12 and 12, you can do that. Really, the possibilities are endless. This is designed to be as customizable, as flexible as you need it to be. So today, let's say I'm going to run 23 all stain samples and 1 unstained sample just for fun. So I'm going to add 50 microliters of my samples, of my panel, sorry. So panel 1 will be my all stain antibody panel, and panel 2 will be, for example, fax buffer or some other kind of buffer. And then I want to incubate 30 minutes, which is pretty typical for most antibody incubations, and it's a good amount of time for settling as well. And I can, quote unquote, put it in the fridge. There are temperature controlled decks, so this will keep the temperature of the plate at the correct temperature while it incubates. Let's say I will wash 9 times again, and I will do a 10 microliter per second flow rate with the same wash buffer. And once again, I can vortex a plate after washing to lift the cells back off for loop number 3 in preparation for the next panel. So this is loop 2. I'm going to confirm loop 2. Everything looks good to me. Continue. It will ask again if I want to enter another step. So you can really just repeat this process. This is now loop 3. Everything looks exactly the same, except I can't use panel 1 and 2 anymore. So you can really just iterate this loop over and over again, all of your steps until your assay is done. So let's say you have a viability stain, a surface stain, and then a secondary antibody for your surface stain, followed by fixed perm, followed by intracellular stain. You can absolutely do all of that through this. All you need to do is iterate the loops over and over again. So this is also easy enough that you can do this at home. You can do this in your office, save the protocol, and then when you get to the lab, just execute the protocol that you've saved. And this will save you a lot of time. This will save you a lot of hands-on time, a lot of pipetting. And when it's done, it will provide the plate to you ready for analysis. So let's continue and just finish up this protocol. Let's say this is my surface stain. I'm going to do 23 all stain and 1 unstained control once again. I will incubate for the same amount of time at 4 degrees Celsius. And a 9-time wash and 10 microliter per second flow rate. So all of these are the same parameters you'll notice from the previous loops. We've noticed a lot of the protocols are pretty simple. You can go anywhere from, there is no maximum to the incubation time, but typically we see 30 to 60 minutes being quite typical. We've talked about temperature already. So all of these you have control over, and it's really not difficult to change the protocol or change some steps in the protocol if you would like to. All it does is just changing a temperature here or changing the number of washes. And so this is really designed to be as flexible, as user-friendly as possible. So let's go ahead and finish this protocol. All right, so I'm going to pretend that I'm done with my protocol now. So I say no more assay steps. Now I have a choice. I can either leave the samples processed in the laminar wash plate itself, and then I can take it to the cytometer if I want to read it directly off of the laminar wash plate, or if I want to store my sample overnight, or if I want to take it somewhere else for another type of processing, I can have it transfer to an output plate. And if I transfer, I can even have it top up additional buffer. So for example, if you're acquiring in 150 microliters, you can have it set to top up so that when you get the output plate, everything is already at the correct volume and you don't have to do anything besides take off the lid when you get to the cytometer. So once this is done, hit OK. You can see the background is processing. It will give you a plate map. So this is how your samples will look. All unique samples are plated vertically. All replicates are plated horizontally. You'll also see panel treatments here. So panel 1 is going to samples 1 to 23. These are the all stain samples. Panel 2 is going to just sample 24 because this is my quote unquote unstained control. So if you need FMOs or isotype controls, this is also a good way to do that. So once this is done, I'm going to check this. Everything looks good. Hit continue. It'll ask if you want to save or not. So let's see, today's a good date, so let's go ahead and save that. 224. So I'm gonna save it as a UChicago protocol for your benefit and in honor of all of you who are here today. So you can hit continue, and then you get to a deck loading place. So we understand that a deck like that can be very overwhelming when you're looking at it for the first few times. And so this is really some instruction. These are designed to be good instructions to load the deck. So whatever is flashing here is the first thing on here. So here's where you would load your sample input plate. And here where it's flashing is where you would load your laminar wash plate. And you can iterate through all of these, make sure you have tips, make sure you have opaque lids. These are used to cover the plate during incubation so as to not photo bleach the antibodies. It will also calculate if you need large volume adapters. So as Ohi mentioned before, these will bring the capacity of every well up to 150 microliters. If you need any, it will tell you how many you need. And in this case, we don't need any, so it'll tell you none. And we've chosen to transfer. It will also calculate and tell you how many output plates and lids you need. So when everything is done, you can just hit continue. And I'm in simulation mode right now. So this is running super fast, as simulation mode usually is. But let me move this timer. The timer is up and you can see how much time you have left. But this is the background of it. This is the trace file that actually will be generated automatically every run. And we haven't really touched on this a lot today. But those of you who are familiar with automation will know that traceability is a huge it's a huge benefit of automation. So with every run that you run through the AUTO1000, a trace file is automatically generated with unique timestamps. It tracks every single movement, every single addition, everything that you could possibly need it to track, it will track. So that when you go back, you can load this into for your records. You can put this into an online lab notebook, so that when the time comes, if you need to go back and see what happened, or if you need to troubleshoot an assay, this will be really helpful come that time. And obviously, this is not in a language that's easy to read for most people, and this is something that we at Curiox can help you with as well. So because we're in simulation mode, I'm actually going to stop the timer. And you can see that this is still, it's pretending to run essentially, because I'm not actually connected to an auto right now at the moment. So you'll see it continue to run, and it will keep doing that until the entire protocol is done. While we're waiting, it will be done in a couple of minutes. Are there any questions right now? All right, I'm going to stop the last timer, because when it's all done, it will give us a final output screen.
I have a question.
Yes.
This is Laura. So what if you have a 20 marker panel? So you need 20 single stains and maybe like 10 FMOs, and you have a viability stain that's on a different step. How do you, because it seems like there are only 10 panels available. Is that your maximum, or are there ways to get more than that, or am I misunderstanding?
Yeah, that is a fantastic question. Thank you, Laura. So that is a question we do get quite a bit, especially now as people are moving into really high color flow, into Cytop, which is typically pretty high color as well. So currently the limit is set at 10 panels per plate, but there are potential workarounds that we can use. And so I'm working on we have a programmer that is currently working on an improvement to extend it to maybe 20 panels. If you need more panels than that, come and speak to us. There might be a workaround that we can figure out. There are little hacks around the system that we can do. And so that's something that we can definitely explore. But the sort of default mode is 10 panels, and we encourage anyone who's interested in more panels to come and speak to us. And we can go through a protocol. If you have a protocol available, that would be really helpful. And we can figure out if there are ways around that in the meantime until we get that sorted.
Also a follow-up.
Does that, sorry?
Follow-up question. So is there a way to skip the wash if you want? Because usually I add FC block and incubate for maybe 5 to 10 minutes, and then I add antibodies, so there's no wash.
Yes.
Is there
yes.
to skip?
Yeah, absolutely. Yes. Apologies, I forgot to mention this. So the incubations and the washes per every loop are completely optional. So you can skip them if you want. If you just enter 0, it will skip that step. So yeah, absolutely. An FC block, wait 10 minutes, and then add something else. That's absolutely doable. Yeah. All right, if there are no other questions, this is a good time to show when the run is complete, it'll give you this screen, and then you can perform your shutdown maintenances, click OK, and then you are good to go. Actually, just gonna run this. Alrighty, and now you're at the very end of your method. So I'm going to stop share. And 1 second. Share this. Alrighty. So we were here just a few minutes ago. So here's just the summary screen. If you are interested and want to request a demo, go ahead and scan our fancy QR code that Ohi generated, and I will hand the virtual stage back to Ohi.
Awesome. Well, thank you, Anne. And at this point, where the floor is open for any other questions that people have. I know we've gone over a fair bit of information. So any questions about how laminar wash works? If you have any specific project in your lab that you're curious as to how the laminar wash can benefit you, this is also the right time to bring up any questions, any thoughts, any ideas you'd like to see, as well as if you'd like to test this out in your lab, we do have demos available. So either scan the QR code, I put a link in the chat box. So people, the stage is yours.